I'm just going to stand for a moment to introduce our distinguished Fireside chat person this morning. It is my pleasure and distinct privilege to introduce and have a conversation with Dr. Rafael Yáñez. He's the professor of advanced therapy at the School of Biological Sciences at the Royal Holloway, University of London. He received his Ph.D. and BSc in biochemistry and molecular biology from the Autonomous University of Madrid in Spain. He leads the Advanced Gene and Cell Therapy Lab at Royal Holloway and is a member of the British Society of Cell and Gene and Cell Therapy, and importantly has recently been appointed editor-in-chief of the journal Gene Therapy. He believes it is very important to engage with the wider society, and I'd like to touch on this morning, and as such is a trustee of the Genetic Alliance of the UK and organizes a yearly outreach event on Rare Disease Day. So I think we'd like to start this morning by, I'm going to hand it over to Raphael and have him talk a little bit about his interests in science and gene and cell therapy, and then also just ask you a few of your highlights, your personal highlights, before we get into a few questions that I have, and then hopefully open it up to the audience for a few questions at the end. So please, welcome.
Thank you, Sarah, and thank you to the organizers, to ARM, for the invitation to speak here. As Sarah has mentioned, I trained in Madrid. I did my biology degree and my PhD over there. I was interested at the time in studying molecular evolution, so evolution at the DNA level, and viruses were an ideal system to study that. So I did a significant amount of DNA sequencing before sequencing technology actually took off. So at that time, we actually trialed some capillary sequencing that ABI was developing. So this is the hard times of sequencing, a lot of Sanger sequencing, but very interesting at the time. We did quite a lot, and the results were interesting. So while I was finishing my PhD in Madrid, I developed an interest in gene therapy. And I moved over to London. That was 1995, and I started working on what at the time was called gene targeting, which eventually became genome editing, and you know the importance of a name for things. So I work on mammalian cells in culture, developing methods to improve the efficiency of genome editing before the advent of the critical reagent, the nucleases that can actually cut the DNA. Delivery of reagents, as you all know, is something very important. So at the time, I wanted to do genome editing in hematopoietic stem cells, and there were no very good ways to deliver that by non-viral methods. So I actually work on the conversion of lentiviral vectors, which, as you know, are very efficient in hematopoietic stem cells, but integrate in the genome into non-viral vectors. And that was a critical development for me, and I believe for the technology, because those vectors, those non-integrating lentiviral vectors, or what we call them IDLVs, for integration deficient lentiviral vectors, retain the transduction efficiency to enter those cells, but they no longer integrate in the genome. So they become episomes that express genes effectively. If the cells do not divide, you get permanent, stable gene expression. If the cells divide, you get a transient wave of gene expression. Additionally, they are actually very good platforms for recombination, either homology-dependent repair in genome editing strategies or site-specific recombination mediated by site-specific integrases or transposases. So the non-integrating or integration-deficient lentiviral vectors complement the efficacy of standard integrating vectors by opening up a number of other possibilities. They become episomal, and they can be used for either gene expression or recombination, or as recombination platforms. On the back of those results, we started working in a variety of CNS, central nervous system diseases. So we use them in the context of eye disease, actually RPE65 deficiency, which is about, we all hope, to become the next marketed product, and Parkinson's disease, the spinal cord injury, lately ataxia telangiectasia. And on the back of that work, again, I became very interested in blood-brain barrier models. So that is something that we are very actively working on. We have a very nice cell culture system to study crossing of the blood-brain barrier based on iPSCs. And we are really keen to develop that, A, as a high-throughput system for screening of potential compounds of all sorts for crossing into the CNS, and B, to develop a 3D system based on flow cells to actually have a proper system to study that. So that's the way in which my lab is progressing. I'm an academic, and I realized looking at the roster of guests that I'm rather avis here. So I'm delighted to have been invited and to have the opportunity of putting my tuppence in this meeting.
Thank you. So thank you very much, Rafael. I have a few questions, and again, we'll open it up at the end for questions from the audience. But in my mind, you sit in a very important position as the editor-in-chief of gene therapy, and quite a privileged and honored place to be, well deserved. You are going to be seeing innovation sort of firsthand as it's coming through in publications and research. As Janet described this morning, we are seeing a number of, through financings, through data, through clinical trials, through now we're going to start to talk about marketed products. The field is moving very quickly. It's become very robust. Janet mentioned the notion of inflection point. In terms of where you sit as the editor of gene therapy and what you're seeing, and I guess we'll continue to see, do you feel like we're at an inflection point? And if so, how come and why?
I do think we are at an inflection point. And everyone can have an opinion, so the question is, what do you base that opinion on? So my own impression is, as you know, it's critical to have marketed products. And we now have a handful. And you could argue that the first few ones had barely any take-up, and we can argue for days about the reasons about that very little take-up. Mm-hmm. But very recently, we have had Spinraza approved, both in the US and in Europe, and that's a different game. It technically may not be a European ATMP or a gene therapy, but actually, in principle, it is. I certainly consider it a gene therapy, and that is meeting a significant need. And we all know that children who would have died of spinal muscular atrophy are now surviving and achieving developmental milestones like sitting unaided and standing that were unthinkable some time ago. And the question is making sure that patients have access to that technology. In the context of SMA, which is a rare disease, we are also in a position where there is a second very promising potential medicine under development, which is an adeno-associated virus vector, serotype 9, expressing SMN, which again is looking really promising. So we are, in the context of rare disease, in a position where we not only have one marketed product, we have another one pushing very hard right behind. That is actually impressive in this context. But that's not the only thing. At the European Society for Gene and Cell Therapy meeting in Berlin earlier in October, there was actually a comment from the current president, Professor Robinelli, saying that for the first time, the number of corporate members was higher than the number of academic members. And obviously, that shows a tremendous interest from the corporate world in this technology. And I think another really important thing is how tolerant the environment has become to significant side effects of gene therapy. So if you think of the case of Jesse Gelsinger, back then it was a really massive problem for the field. But nowadays, CAR-Ts have significant side effects. Some people suffer really significantly from that. And that has not caused a massive problem to the technology. The field is prepared to accept that in those cases, really significant side effects are not a reason to stop development and application. And I think that for me, that shows maturity and robustness in the field. So those are, I think, concrete indicators of how robust the field has become.
That we continue to innovate to solve these problems.
Yeah.
So major advancements in the sector, agree. There are still significant challenges, and I'd be interested to hear what you think are the biggest challenges we have in front of us to continue to, bring gene and cell therapies into mainstream healthcare.
I would say that there are challenges all around, and within the whole spectrum of gene and cell therapy, we need developments. We obviously need further preclinical development. I quote this, I work in the rare disease spectrum. No one really knows how many rare diseases there are. Let's say that there are more than 6,000. At the moment, in England, there are postnatal tests for 9 of those, right? And there are therapies for just a handful of rare diseases. But in the context of genomics technologies, and there is, as you know, a very strong drive in the UK and other countries to apply genomics technologies to people who have either cancer or undiagnosed rare diseases, you can now diagnose maybe 40 to 50% of people with unknown rare diseases applying genomics technologies. So we have more and more people who have a name to their specific disease, but for whom there is no treatment available. And that gap is only increasing because before, in the UK, there is a community called SWAN for people effectively affected by syndromes without a name, SWAN. And that community is expanding because more people realize that they have a genetic disease, and maybe half of those, if a genomic technology is applied, can be diagnosed. But actually, beyond diagnosis, there is hardly anything to offer them. So there are more and more people who have a disease, and there is not that much to offer them currently. And with the reduction in prices of genomics technologies and the inclusion in the context of the National Health Services, that gap is only going to increase. So we need to develop more therapies. Rare disease is, I'm sure you know, but because there are 6,000 rare diseases, about 7% of the population is expected to be affected by a rare disease at some point in their lives. That, in the UK, is about 3.5 million people. And rare disease is still not a research priority for the UK government or for the Wellcome Trust or the Medical Research Council. And I personally think that that should change, that rare disease should be recognized as a research priority. So that's a challenge, to be able to deliver more therapies. We need to invest on therapies so we can actually meet the societal challenge of providing something for an ever-expanding community of people affected with a known disease. So that is a challenge. It's not the only one, and I think the importance of involving all the stakeholders is critical here for many reasons. If you produce something that the patients are not happy to have, they are not going to be massively taken up. The health services are not prepared to offer in practice these medicines because at the moment all we have had really is clinical trials, and that's a very different context. To actually enter the mainstream, you need to bring in the knowledge to those providing the clinical care and the clinical delivery. You have to train the pharmacists in the hospitals to deliver these gene therapies. You have to have the -80 freezers in those hospitals. It sounds basic, but they are not there. All those have to be introduced. So training of personnel across the spectrum is really very important. And finally, and I know that this is the subject of the next Fire chat, so we are not going to dwell on it, pricing strategies are very important. Those of us based in the UK hear every day that the NHS does not have enough funds. And if we come up with therapies that cost between $500,000 and $1 million, nominally at least, per patient, and we think that that is only going to increase in terms of numbers as we develop more therapies, as cancer becomes stratified and the populations in each cancer type become smaller and smaller, this is effectively becoming personalized medicine. If all those cost $500,000 a shot, there is no funds. It's not sustainable. So reducing the prices is critical. And I believe that obviously as the technology develops, as more companies enter the spectrum, prices will come down, but they have to come down very seriously because otherwise we are going to have a fantastic panoply of therapies and an NHS here in the UK and in other countries that simply cannot afford it. And yes, there may be massive savings for the NHS down the line, but if the money is needed today, it's not there. So we need to find new funding models, and I believe the industry has to be mature and realistic regarding what is affordable for the system. Because if it is not, you effectively put the system off. They immediately become defensive, and then it's very difficult to introduce anything.
Thank you. I want to come back to something that you commented on in terms of this group, the SWAN group, those without a name for their indications, and talk a little bit about the approach to rare diseases, and if there are more and more rare diseases with different permutations of these. Couple that with, and I'm going to be optimistic here, to say there are a number of platform technologies, I'm going to call them broadly, from how to treat genetic diseases with vectors or with genome editing or with different types of modalities. As we build these platforms and learn more about these platforms, for example, there's a great deal of information on, let's say, AAV9 now, right, and what its features and benefits and drawbacks are. Do you think that there will be the ability to capitalize on our knowledge of these platforms to then accelerate and be able to use this knowledge to take the next step with these disorders that may fall into sort of similar categories? For example, we need to treat through crossing the blood-brain barrier, or we need to treat liver, and here's just another disorder. Can we put another gene in because we've worked out the vehicle and the delivery? So maybe comment a little bit on how our platforms have come, and can we use those going forward?
So the answer is a resounding yes. I entirely believe that, and I would go further. I think that rare disease is very important for this field, because there is thorough understanding of some rare diseases, and the technology has been developed for rare disease. So those viral vectors that you are mentioning, non-viral technologies, even stem cells, have been developed in the context of rare disease. And the reason for that is that, in principle, if you know what gene is defective, you have a very good idea of the target. You know what to do, in principle. Nothing is simple, but certainly that is a lot truer for rare disease than for common disease. So the technology is being developed and tested, critically tested, on rare disease. Yeah. But then that technology can be applied and will be applied, and my opinion is that the companies that are here for the long game and have the long view is that they are developing technology to apply to common disease, which will be really the money spinner in the field. And as examples, who could have told Oxford Biomedica, just to quote a company, that their knowledge of lentiviral technology was going to be critical for CAR T cells and their strategic alliance with Novartis? No one could. When they started, they were going for CNS indications, Parkinson's, some rare diseases, but they developed the technology, and suddenly someone realized that you could put 2 and 2 together and become something much bigger. But no one could have predicted that. So the technology that is being developed across the spectrum in the context of rare disease, where you know what to target and how to measure, will be applied to common diseases, absolutely. So for me, the clever game is to have, in this context, IP in that technology that you can go on and apply to common diseases when you know what to target, basically. So yes, I'm a firm believer that rare diseases as critical in that context is very important. There is also something else. The patient associations, again, coming back to the stakeholders, very often provide the seed funding for that development. So actually, in this environment, it's not unusual that the preclinical development and even some of the clinical development is developed in the context of patient association funding and even the academic and government environment. And only relatively late in the day, biotech and certainly big pharma come in. So they have been critical. Cure SMA, again, just to give a specific example, provided the seed funding for the development of Spinraza. So it is critical. But yes, just rounding up on this, I think that it's critical to develop the technology because it will be applied to common disease eventually.
Yeah, and I think as in your recent editorial, a couple of very good editorial in September of gene therapy this year, really introducing Rafael as the new editor-in-chief of gene therapy. One of the other areas that you and I talked about, and you commented on it a little bit in the editorial, is not only this notion of technologies and platforms being able to treat additional rare diseases, but really setting the stage for the treatment of and the approach to more complex disorders. So, much of gene therapy, since I began in it 20 years ago, was focused on monogenetic disorders where we're going in to try to correct the gene and get the proper protein expressed in the right tissue at the right time in the right amount. That has been a huge hurdle even to start with, to think about those multi-gene disorders or those disorders that are built on pathways. It seems to me, and as I read and talked to you about that our work in this is really setting the stage for the ability to then address those larger disorders and larger markets for this group. And maybe just comment a little bit on that, or further comment on that.
Yeah, no, I entirely agree that it is critical to develop that technology, that the long-term goal has to be common disease, absolutely. Mm-hmm. And again, I can only go back to the importance of involving all the stakeholders. There are training needs, and we have to address those across the spectrum, everything from health sciences, economic models to introduce this into market, preclinical and clinical expertise, pharmaceutical expertise in the context of hospitals, local production facilities, and I would say reasonable marketing strategies that allow the work with regulators and the introduction of those products in the market.
Yeah, good. I'd like to open it up just to see if we have any questions. I have a couple more, but we've got a few minutes left. If anyone has a question for Dr. Yáñez? I know it's early.
Yeah.
Okay, very good. We have a microphone. If you can wait just a moment. Thank you.
I always tell my students to encourage them. I had to get out of bed as well, so we are all to work here. So there you go.
Thank you. Nick Medcalf, Innovate UK. To your point about the importance of availability at the right price for the therapist, do you see the dominance in that solution coming from appropriate reimbursement and pricing, or from costs of manufacture, or a combination, or something different?
It is a combination, in my opinion. The price has to be realistic. I'll give you examples. I think that's critical. So Spinraza for spinal muscular atrophy is a lifesaver and has been licensed for all types of SMA. But it's only being made available for type 1, the most severe patients. Type 2 and type 3 are not getting it. You are having parents trying to have their type 2 children reclassified as type 1 so they can get access to the drug. And that is with an oligonucleotide, which is not very expensive to produce. So marketing prices is an issue. Obviously, as technology develops and we can make it better and cheaper, then that should have an impact on price. But we need new economic models. The system is saving massively, but the savings come down the line. They are not upfront. So if we demand massive prices upfront, the system cannot cope because it's already at breaking point. So it is a combination of issues. But a realistic pricing policy, I think, is a very important part.
Thank you.
Any other questions? Okay, I'm going to come back to a couple of my questions. So, you began your career in what we're going to call genome editing at this point. Genome editing has maybe a much broader definition now and much more in the mainstream, right? What are your thoughts on that right now with regard to the number of different platform technologies that are there to attempt to edit the genome? And I would just love to hear your thoughts scientifically and sort of looking forward to as, what do you think will be successful?
I think it's incredibly exciting. The times in general are incredibly exciting, and genome editing is one of the platforms that are responsible for that. I do have the sort of long view on genome editing because I started working on that 22 years ago. There were no nucleases. We were struggling to get any genome editing cells. It was possible, but it was very low efficiency. But I like to say also when I teach this to my students that before CRISPR was discovered, there were already clinical trials in the genome editing space for AIDS, CCR5, Sangamo. And there was a Nobel Prize in the field, for the development of knockout technology in embryonic stem cells based on genome editing, even though it was called gene targeting at the time. So the developments do not come out of the blue. CRISPR has been very important. CRISPR has made genome editing democratic. Anyone can do it, basically. It's cheap. The reagents are accessible. Anyone could do it before. It was so demanding that very people actually chose to do it. CRISPR has made it possible for anyone. The reagents are provided at cost. It's very simple to undertake, and that brings, with the number of people working on it, brings massive developments. Only a few days ago, the base editors have been published. So up until now, you had to cut the genome, which we all know has substantial risks inherent to that. But you no longer have to cut the genome if what you are aiming for is to introduce a specific base change. That was unthinkable. And so CRISPR has allowed a massive expansion and has made it available. Now, what models are particularly suitable for that? In general, because still the frequency may be relatively low, if gene-edited cells have a selective advantage of any kind, then they are likely to be taken up first. So I do believe that just following on the experience of a standard gene addition therapy and the clinical experience on hematopoietic stem cells, which arises from the experience in bone marrow transplantation, is an ideal test bed, because actually not only there is significant clinical expertise in that area, but if there are any issues, you do not have to transplant the cells back into the patient. You can analyze them in cell culture, anything you don't like, end of. If you go in vivo, then you lose control, so to speak. So I am as excited as everyone else with maybe a little longer view, and I hope that the in vivo applications of genome editing are considered carefully before we embark into that, because potentially the side effects can be important. And the last thing we want is to put off society and the clinical environment.
On the technology itself.
Yeah.
I think, okay, Laura has given me just a couple more minutes here. And thank you for that on genome editing. I'm gonna ask a pretty traditional last question when having a conversation.
Can I follow up on that
Sure.
conversation you just had? So Raphael, I just want to pick up on the remarks you just made. So obviously we're in a reality now where different nationalities, different countries are regulating research in germline editing of human embryos quite differently. Yeah. And some not at all. And one would imagine, therefore, that various developments will occur. Do you have thoughts on sort of the way the international research community should be addressing either a sort of a framework or any other mechanism to try to establish some sort of common approach, or do you feel that this nation by nation approach is inevitable, or just what will be?
Okay, so I have been to a number of fora of places where the ethical implications of genome editing have been discussed. And the overall conclusion is that genome editing does not bring anything new to the table in terms of ethical concerns, that what applied to gene therapy applies to genome editing. So there is no novelty there. I do believe that we have a social responsibility. We have to act certainly within the law. And at the end of the day, scientists at the bench and in your environment know better than anyone the potential implications. So we do have a responsibility. And in each country, we have to act within our legal framework. What may happen in other countries is a little unpredictable. I think that it's important to lead by example, because if you lose your standards, then you can't go anywhere and say, this is the way it should be done, because they can tell you that's not what you are doing. So I think it's very important to lead by example. We, at the end of the day, if you want to, Western societies have led here. It's very important to retain that moral high ground and demonstrate that what we are doing is according to the law and is acceptable for our societies, and bring people on board and accept that China is becoming a major player here, and we want to work with them, and what we want is to ensure that we work to very similar standards. So yes, I think that international cooperation to ensure that we are all in an even playing field, acceptable to our societies and within the law, is a critical thing. Yeah.
I think we're out of time. I'm going to have to ask the audience, please help me in thanking Dr. Rafael Yáñez for being here. Thank you.